Hi. Okay, in this video I'd like to explain something about the representation of complexes in iRefIndex and interaction data in general. We started with talking about simple binary interaction records where the proteins would have a short label, a type of molecule, etc. And the interaction record itself would have properties like detection method and interaction type. These two qualities are very important when looking at interaction data. The detection method determines the type of interaction that's being implied. For instance, detection methods that use genetics can only indicate a genetic interaction and not a physical interaction. And on the other hand, different physical types of assays are good at detecting different types of physical interactions or associations. And that's the distinction that I want to draw. We can divide experimental methods into basically two different families of methods. The first one would be binary methods, where the output of the assay is binary in nature. So the typical example here is the yeast-2 hybrid assay, where in multiple experiments you use some bait, and depending upon the presence or absence of a signal, you can make a conclusion that some prey protein has actually been interacting with the bait protein. This gives an output that is generally regarded as indicating a direct interaction between the two proteins, the bait and the prey, with some caveats. The other family of experimental types belongs to the immunoprecipitation type experiments, where you take some bait protein, tag it with some tag that can be immunoprecipitated from an extract, and then use that antibody against the tag to pull out the bait plus anything that is bound to it from a cellular extract of some sort, run it out in an SDS page gel, and do MALDI-TOF to identify the components that are associated with the bait protein. The readout from this kind of experiment is what I call associative. The binary interactions between any given pair within this output are unknown. We can't say anything about whether or not F and A directly interact, only that all four of these proteins are somehow associated. There has to be at least 4 binary interactions here, but we don't know what they are. So there are multiple methods that are used to represent these data. Obviously, binary data can be drawn out as a graph or a series of pairwise interactions. However, associative data is a little bit more problematic if you want to put it in a format like MITAB, where you have limited to only defining 2 interactors at a time, A and B, or if you want to somehow combine it with binary data in a graphical view. And there are 3 different methods that are used to do this. The first method is a spoke model, where one of the proteins that was used in the experiment, usually the bait, if it's a bait-like experimental system, is chosen as the hub of this spoke model, and an interaction is drawn between each one of the interacting partners to indicate that each of these other proteins actually has an interaction or belongs to, is associated with the proteins that were pulled out using the bait. The other method is to say, is to use a matrix model, where you take all of the proteins that were observed in the pull down, and draw edges between all of them, or make entries for all of them in a MITAB file, for instance. Both of these spoke and matrix models have problems because both of them are implying or missing potential interactions. The alternative way of representing these data is using something called a bipartite model, where you don't say anything about any of the pairwise interactions, but you're still able to represent it in a kind of a binary-like model. The bipartite model uses a second node type, where each of these round nodes represents a protein, and this hexagon here represents the idea of a complex. And an edge between a protein node, such as E, and this hexagon represents the idea that E is a member of this complex. So in this model, no binary interactions are implied, and it's very explicitly obvious that in the bipartite model that you don't know anything about any of the binary interactions. It's associative data. Okay. So we have to revise our idea of a simple interaction record in that it can be an n-ary interaction record where there are 3 or more interactors, each of which can have short labels and types of molecules, and the interaction itself obviously has a method. In those cases where there are 3 interactors in a record, we are certain that the method that is being used to provide evidence for that interaction record is insufficient to give evidence for any binary interaction. And so the interaction type is almost something like an associative interaction type. So this shows up in each one of the iRefIndex end products in a number of ways. So this is the iRefIndex version in Cytoscape called iRefScape. And here I have 2 nodes. These are proteins, and they both interact with this hexagon here, which is representing the idea of a complex. So you can click on these edges here, and under the Edge Attribute Browser you can find a link out to find all of the other interactors that were also found a part of this complex in this particular experiment. And you can see that the type under the Edge Attributes is an association. It's not a direct physical interaction. And of course, both of these come from the same publication and of course interaction record. Right. If you link out to iRefWeb using the Edge Attribute Browser, you can see all of the other interactors that were found to be a part of that particular complex from that experiment. So this is the iRefWeb version of the same record that I showed you on the previous slide. Here, the source database is intact. Here's the ID. The interaction type is association. This is the PSI-MI code for the interaction type. The detection type or experiment that was used in this case was an anti-tag co-immunoprecipitation, which is obviously only suited for finding indirect interactions, especially when 3 or more interactors are identified. And this is the corresponding PSI-MI code for this term here. So bipartite data or n-ary data also shows up in the MITAB files. With each one of the MITAB files is distributed a README, and in this case I'm looking at the README for iRefIndex version 7.0, and this is the MITAB 2.6 version. And on this page, this issue is described in much more detail, but I'll quickly go over it here. This is what the MITAB file looks like. It's a tab-delimited file. I can import this into Microsoft Excel. And I'm looking at a few of the lines at the beginning. Here, these 2 columns here give identifiers that are unique database identifiers for interactors A and B. In the first few lines, it's obvious that the 2 things that are interacting are proteins, and you would think that these are binary interactions that represent some kind of relationship between these 2 proteins. However, looking down here at this yellow region, there are some things here that are obviously not coming from a protein database. What these are internal identifiers that can be generated by anybody using the sequence of the interactors in this complex. So in the second column, all of these proteins here were identified as being members or associated with some sort of complex. And this is the identifier over here for that particular complex. So just to show you how this looks schematically, in the case of a binary interaction, UID-A and UID-B will point to some protein identifier, and that is giving evidence of some kind of an interaction between these 2 proteins. In this case, it's a physical association. There's a column called expansion in the MITAB 2.6 format And that column there will say none for any case where you're talking about an interaction row in the MITAB file that is indicative of a relationship between two specific proteins. In the case of associative data, we represent, using the iRefIndex file, we use the expansion type bipartite, and you'll see bipartite. And the first identifier will be a complex identifier. This is a hash key of all of the ROG IDs of all of the interactors in this complex. The second identifier, UIDB, points to one of the proteins. So this row here points to one of these memberships describing one member of a complex. So the complex, the entire complex in the MITAB file, will be represented in this case by a total of 4 lines. I want to make one last comment about an important caveat when you're looking at binary data where there's 2 proteins mentioned on the same line in a MITAB file. It's very important to look at the methods. Just because 2 proteins are mentioned on the same line doesn't mean that there's a direct physical interaction between them. And a few examples are given here. I scrolled over to the column called method, and you can see that these first few lines here that all mentioned proteins in the first 2 columns, some of them are referring to genetic interactions. Others, the evidence that was used to provide some sort of relationship between these proteins was actually provided by affinity chromatography technology, which in some cases, if a purified extract or protein is not being used, is insufficient to show direct interaction between things. The other thing that you can look at is also the interaction type column, and you can see that the interaction type is strongly inferred by the evidence type or the method type that was used to show the relationship. So in these first few cases here, these interactions are synthetic genetic interactions defined by inequality. This is the corresponding MI code. One of these is actually a direct interaction, and these others here are indeed physical associations, which is a less strong or less direct type of interaction than this control vocabulary term here, which is direct interaction. And you can see, of course, that all of the interactions or lines that came from the complex entries all have an interaction type of association. Well, all of this is covered in much more detail in the README that is associated with the MITAB files, and you can read more about it in that file on our wiki site. To get to that, you go to the iRefIndex click on data availability via download, and that will take you to the appropriate README. So, other topics that I have yet to cover in this iRefIndex Explained series are iRefScape itself and iRefWeb. These are the people who have been working on iRefIndex, Sabri Razik in my lab, along with Antonio Mora and Paul Bodie, and the Shoshana Wodak group in Toronto.